Thank you very much to Tricia and Yukiko for inviting me. It's been great being here, and I look forward to talking to more of you tomorrow. And yeah, I'm going to talk to you today about the work that we've been doing trying to understand sterile hybrid males in Drosophila. So I'll start by thanking the people in and around my lab who've done the work. This was initiated when I was briefly at Indiana University, where Brian Calvi was generous enough to give me lab space, and I worked with a student who I have no knowledge that I was related to her, and then continued at the University of Rochester, and a large chunk of what I'll talk about was done in collaboration with David Stern at Genitalia. Okay, so my story starts 240,000 years ago when giant flies roamed Africa. And so this is, what I want to emphasize is that this is not very long ago, right? So the parts of the human lineage that would give rise to the Neanderthal and Denisovan populations had already left Africa at this point. And at that time, there was a fruit fly that was living on rotting fruit in Africa. And at some point, about 240,000 years ago, a propagule, an inseminated female, a population of these flies, some number of them got to the remote Indian Ocean island of Mauritius, probably via Madagascar first. When they got there, they would have encountered Rufus cuculatus, the most famous denizen of that island of Mauritius, which would hang out there for another 240,000 years before it was discovered by Dutch sailors and eaten to extinction. About 10,000 years ago, as we developed agriculture and started really accumulating a lot of delicious resources in one place, this ancestral mainland species left Africa, and then probably more recently, with the advent of boats and bringing bananas everywhere, made it across the world, so it is now a worldwide commensal living with humans. 100 years ago or so, Alfred Sturtevant was working with this species and Melanogaster and discovered that they were separate species and named that one Drosophila simulans. And then in the 70s, some French biologists, Jean David and his colleagues, went to the island of Mauritius and discovered the fruit fly there and named it Drosophila mauritiana. So I want to talk about what happened in those 240,000 years and why it is that we think these two things are worthy of being called separate species. So we can consider these two population species of fruit flies genealogically, so they share a common ancestor. There has been some descent. There was some 240,000 years ago. We think this is a pretty clear, simple case of speciation by allopatry. So we can look at their genes, we can look at the lineages, the alleles in these populations, and ask, to what extent do we see separation in terms of their genealogy? So something like this, where this is a case where all the simulans alleles at a locus are more similar to each other, and all the mauritiana alleles at this locus are more similar to each other than either are to alleles from the other species. This describes, for example, the vast majority of genealogies for the human genome relative to chimpanzee, right? So most human alleles coalesce much younger than the divergence time with chimpanzees, with a couple of interesting exceptions like the ABO blood group. This is, in fact, what we see for a minority of loci in the genome for these two species. In fact, what is much more common is a situation like this, where some simulans alleles are more closely related to some mauritiana alleles than they are to other simulans alleles. And that reflects the fact that the ancestral population of these two species was quite large. The extant population of simulans is quite large, so there's a lot of polymorphism, a lot of segregating variation, and so there's a lot of incomplete lineage sorting. So these things just haven't sorted out yet. And in fact, if we estimate the average age of the within species genealogy, it's about 2 to 3 times deeper than we infer the species divergence time. So for most of the variation in each of these species, it's segregating and it is shared with the other species. So the take-home point is they're not very different at all at the DNA sequence level. Their divergence, pairwise divergence, is about 2%, and that's sort of, again, on par with levels of polymorphism in each species. We can also ask, we think that that was an allopatric event. Have they been separated by geography ever since? And so one way to ask that question is to look for evidence of introgression. So can we look at genome sequences from these species and see evidence that there has been recent gene flow? And so the way we decided to do this was with a statistic called GMIN, which I like because it's sort of simple and intuitive. There are a lot of fancier, more powerful methods out there as well. This method just looks at pairwise distance between sequences from two species and calculates a statistic, which is the ratio of the minimum dxy, so the two sequences from the two species that are the most similar, to the average of all of the pairwise comparisons. And so if you have a recent introgression event, that would look like this, where a lineage from one species invaded into the other species. And so your minimum dxy will be small, particularly relative to the mean. And so using this statistic as a metric with sliding windows across the genome, we do indeed find evidence for recent introgression. And I will say this one feature of this statistic that's sort of a benefit of its low power, benefit whatever, is it only detects recent introgression. So what we're seeing here is likely something that happened very recently. We can't use this method to really detect older introgression. So we can see if we plot in either 10 or 5 kb windows, we can see, for example, on chromosome 3R, these regions where we see significant evidence for introgression. And in most cases, we see a bunch of contiguous windows that show that signal, suggesting that the introgressed segment is bigger than 5 or 10 kb. And if we look at a genealogy of sequences from a locus that shows a significant GMIN, it looks something like this, right? So here are 10 Martian alleles, and they're clearly very closely related to these simulans alleles, and these simulans alleles are more similar to these Martiana ones than to other simulans alleles. So across the genome, we find evidence for 48 regions like this. Each of these would be, that's a region, that's one very small region, that show evidence for recent introgression. Probably, I don't know, but I'm going to speculate that that's probably due to simulans getting to the island of Mauritius via human transport. They're not very divergent, and we see evidence for recent gene flow. So this is a case of speciation in the face of introgression and gene flow. We call them different species in part because they do have a few slight morphological differences. The main morphological way that they can be distinguished is the male genitalia. So here's the back end of a male. They have structures that are involved in mating, involved with interacting with the female and grabbing onto her, claspers, a posterior lobe, and an anal plate. These things have bristles on them. And you can see that the shape of the clasper is quite different between Ma and Sim. The shape of the posterior lobe is quite different, and the shape and sort of arrangement of bristles on the anal plate is also different. And this has proved to be a pretty rich source of study for Evo-Devo for a number of labs, under trying to map the genes that are involved in these differences. And it probably has an effect on mating. Females tend to have structures that sort of match male structures. We know that they are also divergent. They have some behavioral prezygotic reproductive isolation. Flies sing songs, males court females, and these two species sing rather different songs. Females are not always choosy, but they can be choosy, and they can discriminate based on song. There's also some divergence in cuticular hydrocarbon profile, so they probably smell different to each other. But the main reason I call them different species is because there's very strong postzygotic reproductive isolation between these two species. So in either direction, when you cross them, the hybrid F1 females are fertile and the hybrid F1 males are sterile. So this is presumably a barrier to gene flow in nature and helps keep them separate species. I will just point out that this observation, I'm going to focus on these species, this cross, and dig down into this system, but we'd like to think that this is, what we're learning is informing broader patterns. And indeed, the pattern of hybrid male sterility is a very famous pattern in evolutionary biology. In flies and mammals, it's part of Haldane's rule. So Haldane's rule is the observation that if you have asymmetric incompatibilities, that is, in a cross where only one sex is inviable or only one sex is sterile, that sex is preferentially the heterogametic sex. So in mammals and flies, where males are XY and females are XX, that means males suffer the incompatibility. In birds and butterflies, where females are XY, ZW, and males are XX, the females suffer the incompatibility. So this really shows that it's not about male versus female per se, it's about having differentiated sex chromosomes. And you can see from these numbers, Haldane's rule is a profoundly robust rule. Like, this is one of the strongest patterns that you'll see in evolutionary biology. And we know from some really clever work done by David Presgraves and Alan Orr in mosquitoes that these are the most amazing papers because all they did was read some literature and came up with these amazing ideas, that the mechanism underlying Haldane's rule for inviability and for sterility seems to be different. So the way that they did this test was they had data on a bunch of hybridizations in two lineages of mosquitoes, Anopheline mosquitoes, which have heteromorphic X's and Y's, so the Y chromosome is very different from the X. Most of the X is homozygous. And Aedes mosquitoes, which are also XY, but the X and the Y are very similar. So under the microscope, they look the same, and they probably share many homologous genes along a lot of their length. So in these two groups of mosquitoes, you see quite different patterns with respect to Haldane's rule for inviability. So you can see in Anopheles, you see very strong evidence for Haldane's rule out of 24 hybridizations that show asymmetric lethality. 21 out of those 24, the males are dead, and the females are, only 3 out of them are the females dead. However, you see much less of that in Aedes. So you see most hybridizations, if there are lethal incompatibilities, they affect both sexes and an equal, and it's 1, so it's small, but equal number affecting males and females. So the interpretation there is that lethal incompatibilities are recessive. And so a hemizygous X chromosome exposes those incompatibilities, and so now those individuals are dead. Those incompatibilities, then the hypothesis goes, are masked in Aedes crosses because the X and the Y are mostly carry the same genes, and so those recessive incompatibilities can't be exposed. However, if you look at the sterility-causing incompatibilities, you see that Haldane's rule is strongly obeyed in both Anopheles and in Aedes. So in this case, because males of both lineages show sterility, regardless of whether they have a heteromorphic or a homomorphic sex chromosome, the interpretation is that it's not recessivity per se that drives Haldane's rule for sterility, but it's that sterile incompatibilities in XY taxa are male-specific. And so I think, going forward, one big question that needs to be answered is, to what extent is this can we do these genetics that I'm going to talk about in a ZW system to see if that is also true for birds and butterflies? Okay, but the other pattern that, so I've got here 2 rules of speciation. Haldane's rule is one of them. The other pattern that has been demonstrated in Drosophila and in mice and in other species is the large X effect. And so this is the observation that when you can map incompatibilities to regions or loci, they tend to be strongly enriched on the X chromosome. And so the X chromosome seems to be a hot spot for the evolution of these incompatibilities. So this is a figure from a study showing the location of introgressions from one species into the other that cause either lethality or sterility. The red ones cause sterility, and you can see that they're strongly enriched on this Drosophila X chromosome. All right, so we've got this cross, these 2 species. They form fertile hybrid F1 males. Females, males that are sterile due to these sterile male incompatibilities. And so the question then is, what are these, and how many of them are there, and how do they work? So previous work that's been going on for a while now, mapping these incompatibilities in this cross, has come to the conclusion that there's a bunch of regions, at least 15 regions, where you move material from Mauritiana into a simulant's background and males are sterile. And about half of those 15 regions are on the autosomes, and about half of them are on the X chromosome. However, the X chromosome is only 20% of the genome, so here you see that enrichment, the large X effect. All right, so the questions that we're trying to address are, how do these incompatibilities cause hybrid male sterility? What are the genes that underlie these incompatibilities? And what are the evolutionary forces that drove all of this hybrid sterility in such a short amount of evolutionary time? And I'll start with this question: how do they cause hybrid male sterility? And I'm going to approach this question in 2 ways. One is, what are the genetic interactions that do this? And then what is the developmental nature of the sterility? So this is the system we work with. We have a bunch of genotypes of Drosophila simulans where we have backcrossed little pieces of the X chromosome from Drosophila mauritiana. They're marked with visible markers so we can follow them and we can make sure that every generation this remains intact. They tile over 80% of the euchromatic portion of the X chromosome. All of them cause males to be sterile. All right, so in here is a factor or factors that are incompatible with the simulin's genome and cause sterility, and in here, and in here, and in here, and in here, and in here. All right, so these regions, these factors, whatever they are in there, cause sterility by interacting with alleles in the simulin's genome, right? This piece of an X chromosome is fine in Drosophila marciana. This male is totally fertile. But when we put this piece into a simulin's background, it now causes sterility, so it must therefore do so because of these interactions. And these interactions could be between X chromosomal and autosomal alleles, or they could be between X chromosomal and Y-linked alleles, for example. It's been known previously that the Y chromosome can cause sterility, suggesting the evidence of XY interactions. So these are the movements of Y chromosomes that have been attempted because they're possible because of the reagents we have. And so it's known that the simulin's Y is sterile in a Seychellia background. I should say, for those of you who aren't aficionados, Drosophila Seychellia is a third species that diverged by about the same time from simulins and marciana as they did from each other. So there's a polytomy there. And Seychellia is also endemic to an island in the Indian Ocean, the Seychelles. So we've got two island species and then a cosmopolitan. So the only fertile Y chromosome that was previously reported was you could move the Y chromosome from Seychellia into simulins and have fertile males, but it was shown that, or thought that the simulin's Y was sterile in a Seychellia background, and that in either direction, moving the Y chromosome from sim into Ma or from marciana into simulins caused sterility. However, I was reading those papers and realized that this paper that did this test, they said the marciana Y is sterile in simulins, and the evidence for that was less than 5% of F2 males have motile sperm. So that's a great definition of sterility, but I read that and thought, so you're telling me there's a chance. And so we took a chance. And so we used these compound X and compound XY chromosomes that we have in simulins to do these crosses. So we backcrossed, the details don't matter, but the point is we did this in such a way that we were looking for F3 males to be fertile, and they, by and large, were not, but we found a very, very low rate of fertility, and this was only made possible by a super-duper undergrad who was like a fly sensei. And so he was able to get flies out of this, and indeed, we found after many generations of backcross that this marciana Y chromosome is not sterile in a simulin's background. So here is our control simulins. These are the number of offspring per male. So we have a standard fertility test we run males through. We give them 3 females, let them go for a week, take them out, see how many progeny he produced. And so here's our simulins control. Here's our marciana control. Here's our YMA strain. So the genome is all simulins except for the Y chromosome. You can see that they're not equivalently fertile, but they're definitely not sterile. So we think that, that F2 sterility was probably segregating autosomal stuff that hadn't been cleaned out, but that in fact, in our hands, this marciana Y is fertile in a simulin's background. It's not without problems, so it does seem like it's, for example, a lot more temperature sensitive than the simulin's Y chromosome. So if we take our pure sim and raise them at 3 temperatures, so this is the developmental temperature. We do all of our fertility assays at 22 degrees. We see a pretty standard pattern for ectotherms where at high temperatures males produce more offspring. But our YMA shows this really striking narrow range of fertility where they're pretty okay at 22C at room temp, but you move them down just a couple degrees or up just a few degrees and they become almost sterile. So we're sort of lucky that we've been doing stuff at room temperature. So not sure what that means at the moment, but it does suggest that it works, but it doesn't work perfectly. So, but the fact that we can get these males and they produce some offspring means that we can now do this test. We can ask, for any of our X-linked regions, do they interact with the Y chromosome? Because this genotype is sterile, but we can now put in, because we know there are no serious, totally sterilizing interactions between the marciana Y and the rest of the simulins genome, we can put in the marciana Y and ask, does it recover fertility in any of our introgressions, which would suggest that there was, that sterility was caused by an XY incompatibility. So we did that, and the answer is no. We see no rescue by the Y chromosome. So here are our 2p intervals with the simulin's Y. Here's our 2p interval with the marciana Y. You can see some of these are not completely sterile. Under optimal conditions, we get some low level of fertility, although some of them are completely sterile. When we add in the YMA, things are either the same or get worse. So that tells us that all of these pieces of the X chromosome must cause sterility by interacting with autosomal factors. We've ruled out the Y chromosome in these cases, with a caveat that I will come back to. Okay, so these incompatibilities cause sterility by interacting with autosomal alleles, not the Y chromosome. We can ask what goes wrong during spermatogenesis. So to do that, we look in hybrid testes, and all of our interactions seem to converge on a rather similar phenotype, which is a late block that results in the production of no sperm in the seminal vesicle. So it's not that they make sperm, but the sperm don't work. They don't finish making sperm. So here are pictures from a wild-type simulans. So here's a Drosophila testis, germline stem cells. Spermatogenesis proceeds more or less like this. As 64 spermatids elongate in concert, which is the way flies do it, you get these nice bundles. So these are 64 sperm tails all lined up. And you see these here. And then we can mark DNA and see the nuclei reshaping. So spermatids after meiosis are round. Sperm heads, the nuclei are needle-shaped. And what you can see here are nuclei, 64 of them that are reshaping together. The final stage are these kind of tight sort of lozenge things. And then what we're seeing here is a signal of protamines. So protamines are histone-like proteins that replace histones in packaging sperm head DNA into sperm heads. And this is a transgene where the protamine is labeled with GFP. And so we can see these nuclei they put on protamines, so they've got protamines on their DNA now. They're elongating, the nuclei are reshaping, and then in the seminal vesicle, we can see mature sperm. So each of these little green lines is a single sperm head. All right, so that's our wild type. I would say all of these pictures were done by a technician, Kathleen Gordon, in the lab. So this is one of our sterile phenotypes, one of our sterile genotypes, and we see this late sort of problem. So we see bundles. So to a first approximation, the bundles seem normal, they're there. We see nuclei reshaping, at least starting to reshape. We never get to this like nice sort of tight finished phenotype. They seem to stall out earlier than that. We never see any sperm in the seminal vesicle. So the last step, that individualization seems to be required for, or at least happens sort of concurrently with moving into the seminal vesicle, so they don't do that. But we do see that they incorporate protamines. So there's that stage, they get to that point, they reshape, they start to reshape, but they never finish. So there's some block there. And this is true of all of our different regions. So to a first approximation. So 2p1, 2, 3, 1, 3, 4, and 5 all show the same kind of phenotype with this late block. They have sperm bundles. They fail to individualize. The proximal most introgressions 6 and 7. In addition, they seem to show these sort of phenotypes where the nuclei come apart and sort of spread throughout the testis. These ones don't seem to reshape much at all, but they still incorporate protamines. So we seem to have a common developmental arrest kind of a phenotype. It's not clear at the moment whether that reflects something about the evolutionary process or whether that reflects something about the developmental process. In Drosophila melanogaster, a large fraction of male sterile mutations cause phenotypes that are grossly similar to this. So that suggests that this may be a stage at which, if something goes wrong earlier, this is the stage at which we can see it, the first stage where it's visible under the microscope. I will say that we've started to look at the structure that does individualization. So there's an actin-rich structure that starts at the head and progresses towards the tails to individualize those sperm. And I don't have a picture because it's very preliminary, but it seems like these genotypes may not assemble that structure. We are having trouble finding it. So that may be one of the reasons why they don't individualize. Okay, so these incompatibilities cause sterility. These X-linked alleles, by interacting with the autosomes, they cause this azospermia phenotype, this late arrest, this failure to individualize. We'd like to know, ultimately, what are the genes that are responsible for these incompatibilities. And here I will tell you how we've been going about this and ultimately not give you any real information on this. So to do this, we do just genetic mapping. We take each of these intervals, and we just use recombination in one generation to break it up. So here's our interval. We break it up with one generation of recombination. We do this with another transgene that allows us to trap this recombination breakpoint. So this is a now stable stock that we can keep as long as we want to do phenotype and genotype analysis. And all of this is done with these visible markers that affect eye color. So if you've got both P elements, you've got red eyes. If you've got one or the other, you're orange. You've got none, you're white. And then we flip on one of these YFPs, and so you get this orange YFP fly right here. So you can really crank through this, and we did. And so we generated about 600 recombinant lines that we genotyped, and then we phenotyped, and then we genotyped by light shotgun sequencing. And this is what we found. So in this plot, each of these bars indicates the extent of an introgressed Mauritiana segment, as inferred by the sequencing. And the color of the bar indicates the fertility. So the yellow ones are sterile, and the blue ones are fertile. And so you can see there are sort of roughly 3 big fertile zones where you can introgress the distal, maybe 5th or 6th of the X chromosome with little loss of fertility. This middle bit seems to be compatible with fertility, and this bit here. Everywhere else, we see either evidence for strong factors, so we think that these are regions where there's likely a Mendelian-ish factor or a bunch of tightly linked ones that cause sterility, and this is where we've been focusing a lot of our effort. But we also see a lot of quantitative effects suggesting that maybe there are multiple things in here, each of which partially sterilizes males. You may also recognize that in here we have this big sterile zone. We have a couple of rare introgressions that seem to be fertile, but that's probably error. We can't really get inside here because our scheme, we always bring in Mauritiana material from this P element or from this P element, and so there's no way to get in the middle here without including these flanking bits. And we know that there's sterility in these flanking bits, so the sort of terra incognita in the middle here, we don't know how many more factors are in there. But in trying to walk in and map these factors, we've gotten into this region, and we found something that we weren't expecting, but we probably should have been, which was a hybrid sterility factor that had previously been described in this cross. So we have rediscovered, confirmed Odysseus, which was described as a hybrid sterility factor by Chengyi Wu back in the 90s. But it's a pretty strong one, so basically, here's the Odysseus locus. And we're bringing introgressions in from the right here. And if the introgressions stop short of Odysseus, males are fertile. And if they include the 3' exons of the gene, which include the homeobox domain, this is a homeobox gene, then they are sterile. So this seems like a pretty strong sterility factor. And one of my graduate students has been working on this, trying to understand more about Odysseus. After Chengyi Wu published this, there was a lull for a number of years, and then Harmeet Malik did a really cool study on Odysseus where they were trying to understand the mechanism of sterility and discovered that Odysseus has very interesting and different binding properties between the two species. So homeobox domain, it binds DNA. What they found was that the Odysseus protein from Mauritiana binds to the Drosophila simulans Y chromosome, but it does not bind to the Drosophila mauritiana Y chromosome. So it doesn't bind to its own Y. So here we have chromosomes from larval brain cells. And so this is D1 marks repetitive DNA. You can see there's the Y chromosome. And then this is a tagged allele of the simulans Odysseus. And it binds a little bit to its own Y and to the X. Here is the, here is again, the simulans allele. Here it's not binding to its own Y. This is the Mauritiana allele, and you can see binding to the Y chromosome as well as to the fourth. So Odysseus simulans binds to X and fourth, Mauritiana binds to the X fourth, and the simulans Y chromosome. And so here's, in hybrids, they looked and could see that Mauritiana Odysseus does not bind to the Mauritiana Y, neither does Odysseus sim. And so they summarize their results in this little cartoon here. So again, the Odysseus protein seems to bind to repetitive DNA, which is enriched at the base of the X on the Y chromosome and the fourth chromosome. So the simulans allele binds to the simulans X and the simulans fourth. It binds to the Mauritiana X, but not the Mauritiana fourth. The Mauritiana allele of Odysseus binds to the simulans Y and the simulans fourth, but not really its own Y or its own fourth chromosome. So this binding pattern suggested that maybe the cause of sterility with an Odysseus protein from Mauritiana is binding inappropriately to the Y chromosome. It seems like the effect of Odysseus binding tends to be for chromatin to be decondensed. And so the idea here would be maybe it is inappropriately decondensing the simulin's Y chromosome, causing sterility. Now, all of these images were taken in larval brains, right? So we don't actually know if Odysseus A is binding to the Y chromosome in the testes, and B if it's decondensing the Y chromosome in the testes. So we didn't test that, but what we did test was whether genetically we can see evidence for this Odysseus Y chromosome interaction. And because we've got our Odysseus Y in our Mauritiana Y in our simulin's background, we could do that. So we have here our background simulins. Here's our Y-moss strain. Here is our Odysseus introgression, which is incredibly sterile, not 100%, but out of 46 males, we had one who produced about 20 offspring. And lo and behold, if we introduce the Mauritiana Y chromosome, we do indeed recover some amount of fertility. So this is genetic evidence that, yes, Malik was right. The interaction, it seems to be between Odysseus and the Y chromosome, and that binding that they saw in larval cells is likely part of the story of sterility. Now, it's not a complete recover of fertility, so it may be that there are other places that Odysseus is binding. We haven't tested the fourth chromosome, for example. So there may be more to it, but we do see evidence for this interaction. So this would be an exception to what I stated earlier. I said there are no XY interactions causing sterility except for Odysseus. And the reason that we don't see that with our bigger 2p6 introgression is presumably because it's got Odysseus in it and a bunch of other things, and those other things interact with the autosomes, and so they are not rescued by replacing the Y chromosome. Okay, so what are the genes? We don't know yet. But we're working on it. We've refound Odysseus, and we're trying to map some of those other factors that show strong effects on fertility. So the last thing I want to talk about is trying to understand what are the evolutionary forces that fix these changes. So how is it that in 240,000 years, these genomes have accumulated changes that make them sterile 15-fold over only in males? So there's been a lot of ink spilled on this idea. There are way more ideas than there is data. And so there are at least, these are only sort of the most prominent models. There are other models as well as to why you might see X-linked hybrid male sterility factors evolve so rapidly. So faster X evolution, and the way I've done this is who proposed the idea, and this is evidence for the idea. This is the idea that recessive alleles, beneficial recessive mutations will fix faster on the X chromosome because they are exposed in males. So the evidence for this is kind of thin. It's just that Odysseus seemed to have evidence of recent positive selection. I'll skip this one and come back to it. X chromosome misregulation is the hypothesis that X chromosomes have special regulation in the male germline, and that regulation may be disrupted in hybrids, leading to sterility. That seems to, for sure, be a part of the story in mice. So in mouse male hybrids, they see massive upregulation of the X chromosome. And then gene movement, definitely there's an example of it. So this would be a gene that over evolutionary time moves to a different chromosome, and so then if you make a Backcross hybrid with the right combination of chromosomes, it will be lacking the gene entirely and will be sterile. And so there's evidence of that from Drosophila, but it's unclear how general it is, given rates of what we know about rates of gene movement. It doesn't seem like that process happens fast enough to account for all of that hybrid sterility. So the second one is meiotic drive, and so this is the idea that X's and Y's are engaged in a conflict in the male germline. If an X chromosome can do something to impair Y-bearing sperm, it will increase its frequency in the next generation and get a selective advantage. The same is true for the Y chromosome. If it can somehow impair X-bearing sperm and gain an advantage, that is favored. That's true of autosomes, but autosomes are largely the same across their sequence, so it's very difficult for them to evolve a system that can detect, can distinguish self and other. But X's and Y chromosomes that are very divergent in their sequence, that should be relatively straightforward. So we think that meiotic drive should be enriched on sex chromosomes. And so then how would that work to cause speciation? Well, you could imagine an X-linked distorter arises in a population. So this is an allele that kills Y-bearing sperm. It will be favored, but obviously the increase in frequency of that allele comes at a great cost to the population in two ways. First of all, males that are killing half their sperm may have a loss of fertility. They may be lose out in sperm competitive ability. And so alleles on the autosomes that can suppress that distorter may be favored. In addition, if that comes to high enough frequency, the population may become female biased, and so now there's a selective advantage to producing sons. So again, that will put pressure, selective pressure for autosomal alleles to suppress that distorter. And obviously, if the Y chromosome can evolve to escape this distorter, that would be favored as well. So we can imagine that there are autosomal suppressors that evolve to silence that X-linked driver. Or we could imagine a resistant Y. And so that scenario sounds pretty consistent with what we observe for Odysseus, right? So we have an X-linked gene that binds to the Y chromosome, but it seems like so the Mauritian allele of Odysseus binds to the simulin's Y, but it doesn't bind to its own Y chromosome. So that invokes a story where at one point the Mauritian allele of Odysseus evolved an ability to be a driver, and then to escape that, the Mauritian Y chromosome lost the sequences to which Odysseus binds. So, that's a story, but it seems like a plausible retelling of what may have happened with Odysseus. We have other evidence that meiotic drive may be associated with sterility. So the example I put up there is identification of a gene in Drosophila pseudoobscura that seems to, sort of the magic gene, do both. Both is a hybrid sterility factor and a driver. Other evidence for this comes from, again, Drosophila simulans and Drosophila mauritiana, where long ago Yun Tao mapped an autosomal sterility factor that he named Timmy because it, in addition to being a sterility factor, caused sex ratio distortion, so an excess of daughters, so too much yang is what Timmy stands for. And very recently, Eric Lai has discovered that Timmy and a nearby related suppressor called NIMI encodes a small RNA, and that small RNA suppresses an X-linked distorter known as, or a pair of them, family of them, known as DOX and MDOX, so distorter on X and mother of DOX, because they're duplicates. So Timmy is formed by some, we don't really know exactly, some crazy duplication rearrangement madness, where it now produces a small RNA that maps to DOX, and if you get rid of Timmy, DOX and MDOX are unleashed, and now you have an excess, 90% daughters. So if this is the case, right, if Timmy is a suppressor of DOX and Timmy is a hybrid sterility factor, well, maybe DOX and MDOX are hybrid sterility factors. Are they hybrid sterility loci? And so I'm not sure my lab appreciates how much I put up cartoons like this that seem, maybe if you're a young graduate student, this seems super bleak. I just think this is funny. So we've now been producing detailed tributes to the wrongness of this idea. So DOX and MDOX are not, unequivocally are not hybrid sterility loci. First of all, DOX and MDOX sit right here in the middle of this fertile zone, so clearly they're not causing sterility. And secondly, of those 48 introgressed regions that we found with that scan, 47 of them were on the autosomes, and 1 of them was X-linked. So that's a significant depletion on the X chromosome, and that's been seen in many species. Sex chromosomes seem to be refractory to introgression, presumably because they carry hybrid incompatibilities. And so introgressed DNA can't escape from those linked incompatibilities. So we only find this one region on the X. It looks like it recently introgressed, and of course, that's where DOX and MDOX are. So not only are they not hybrid sterility loci, but these selfish genetic elements recently moved between species. We don't yet know the direction, although, again, given sort of biogeography, it was likely from simulans into Mauritiana. But in fact, this genealogy that I showed you earlier is the genealogy of the MDOX locus. So you can see these are almost identical. DOX, similarly, we see evidence for Mauritian alleles that are much more similar to simulans alleles than other simulans alleles are to their own species. So in conclusion, it may be the case that meiotic drive can sometimes promote speciation, can be a driver of male sterility causing hybrid incompatibilities, but it's not that simple. They can also jump species boundaries, and if you think about it for a second, that makes good sense given what they do. If they can invade just like any other selfish element, like a transposable element or a Wolbachia, if they get into a species and they're not suppressed, they can take off. And indeed, in this example, they jump species boundaries, and in this small region of the genome, they've homogenized the genome. So they've sort of locally undone any speciation that might have been there and made this region identical between those species. So, I think ultimately really understanding this question, we need more of these genes, and so we're hoping that by identifying other sterility factors and the actual nucleotides that changed, we can then sort of develop the story of what's going on. And it may be that each of those models are correct, and that the very unsatisfying answer is there's a lot of different ways to get rapid evolution of hybrid male sterility. All right, and so I have gone super fast, but with that, I will stop and take any questions you have.
Incompatibility factors. Do you think that's just kind of the gradual accretion of one at a time? Do you think there's more sort of commonality, some sort of shared mechanism?
There's no way it's gradual, because it was just too short of a time. That's a great question. I don't know, but you'd like to imagine, like, did 10 different things happen, or did one thing happen and it took over? Right? So, we have great genome sequences for these species. It's not the case that, like, there's an obvious transposable element that's sitting, under each of these. But I imagine that there must have been, a system or something that proliferated, or that, yes, there must be some common shared cause. Also, the common developmental arrest suggests a common pathway, potentially. So, I think at the end of, if we can solve this mystery, it's going to be like one thing or a few things.
And these were all interacting with the same introgressed region on the X to start with, or did I miss that? You introduced, I think it was a region on the X.
So we have all these different regions on the X. Oops, this will do.
Sorry, okay. I was thinking it was the autosome factor that you were back.
Yeah, so that's the, do we have, I don't have it here. Anyway, that's the whole, like, other half of this, right? That's the one thing, is that this is only half of the story. So, it would be great. I would love to start trying to map the autosomal partners that each of these work with. So I guess we know from earlier work that there are a bunch of sterility factors on the autosomes. Where are you? Oh my God. Nope. There. Now, these are almost certainly not what, well, they could be what these are interacting with, but they also could be interacting with other autosomal things, or with the Y chromosome, or with other pieces of the X. So we've got 15 regions, and then somewhere between 15 and 15 choose 2 interactions. So the bad news is you could spend your whole life doing this.
Odysseus interaction with the Y, melanogaster, the deletions that occur on the Y, do any of them phenocopy the spermative development defects that you see? Say whether the regulation of an individual gene or whether the deletion, there's phenocopying between melanogaster.
So my understanding of Y effects on melanogaster is many of the sterility factors are protein coding genes. So you delete those, do those males, does an KL2 mutant make sperm, but they just don't work? Is that?
Those are not necessarily like Odysseus or that kind of genes, right?
No, I know they're not. So I think, I don't know of, and I think if you just take away the Y chromosome at all, then you have, massive misregulation of these stellate genes, and that phenotype looks earlier. Am I right about that? Maybe.
There's even that we don't understand. I mean, if you get rid of the Y because Y chromosome has lots of
Right, right.
activity genes. But then certain aspect of it, I mean, like, you just mentioned like sterate, right? Suppressor of sterate. If you just get rid of that suppressor of sterate or the pyruvate pathway, it's worse than
The whole Y.
not having a whole Y chromosome. I mean, we don't understand.
I think the short answer is I don't know of any Y-linked manipulations in melanogaster that phenocopy this.
Is X chromosome enriched with genes involved in spermatogenesis? I would have thought it's opposite because I remember X chromosome is silenced during.
So in flies, the X chromosome is neither silenced, nor is there a depletion of spermatogenesis genes. There is, as far as I can tell, there are exactly the number of testis-specific genes on the X chromosome that you would expect given by how big it is. So it's neither enriched nor depleted for genes involved in male sterility, or expressed in spermatogenesis.
Has the effect of Odysseus been measured in multiple genetic backgrounds in simians? Or
So we are re-introgressing different Ys to see. We've only put one Martiana Y into one simulan's background. Sorry, you asked about Odysseus. Well, we've also only done one Martian Odysseus into one simulan's background. I don't know of any studies looking to see if there's variation, polymorphism in that. I know Odysseus shows a fairly high number of substitutions between species. I don't know how polymorphic it is. That's an eminently answerable question.
Is there a difference in the repetitive sequences of the Ys between, I can't remember what the directions of the populations were, that it's thought to interact with versus not interact with the Odysseus?
The Y chromosomes in these species, yes, there is difference in the repetitive sequence. There's also difference in a lot of repetitive sequences on the X chromosome. And so that's something that we're just starting to think about. But so these simple repeats, these satellite sequences are, first of all, about 30-fold enriched on the X relative to the autosomes in both species. And so they're also enriched on the X in these species relative to melanogaster. And they're highly dynamic. So some of these satellites have gone bananas in Martiana. Others of them have expanded in simulans, and some of them are in different places. So that's got a really strong appeal because these are sequences that are very dynamic. So now you've kind of got maybe this is what happened, is these satellites, however they are turning over, maybe they're doing it fast enough to account for this. And we're learning more and more that they have an important role in male fertility in flies. So I think that is where we are turning our eye. But I can, obviously that's not the whole story because Odysseus is not a satellite DNA, that's a protein coding gene. However, maybe that's part of the story because it's binding to those kinds of sequences.
Is there a good idea? Do you know much about the binding motifs of Odysseus in general? I mean, I know it's difficult to sequence the Y chromosomes anyway, so it's a bit of a long shot. But do you know what they bind?
No. And so that would, it's got a homeobox domain, but that also seems, eminently doable.
A little bit of off-topic question, but as you probably know, there's a lot of interest, theoretically at least, in using myric drive as a form of biological control or extirpation or extinction. So based on, although this is not the main aspect of your research, but based on your understanding of this particular system and its extrapolation, do you think it's possible to design a safe mechanism of myric control, or would you say it's just a bad idea?
I think it's appealing. I think it suffers from the same problem that all of those approaches suffer from, which is, or all of the sort of engineering, a drive, whether it's, meiotic drive or not, which is populations are going to be resistant. There's going to be variation out there in the strength of resistance, and by putting this in, you're going to select for those alleles. So, I don't know if that's dangerous. It might just not work. Now, certainly the Wolbachia 80s stuff, has actually been very successful in mosquitoes. So that, argues against my concern.
I was concerned mainly with the leakage of this factor outside of your target populations or species.
I mean, given that these species hybridize and species everywhere are hybridizing, certainly yes. I mean, that has to be. I think if one is designing one of these systems, you have to assume that there's some rate at which it may spread into related wild populations. Now, maybe that's less of a concern for something you've engineered to negatively affect your organism, in contrast to things like herbicide resistance, or insecticide resistance, which benefit the recipients. But a driver or something that's sterilizing males, yeah, it might spread, but it's not going to get very far if it actually has the effect you want it to have. I think the bigger concern is that the species just, evolve around it.
So, these, the sterile regions, and then you have actually this clearly sterile male. Can you simply sequence, what genes are regulated or misregulated or whatever, to see the actual, the direct cause of the sterility, to see if, right, because clearly this, the inter-specific program comes down to something very, very specific. And by knowing which genes are regulated and misregulated, you might be able to get to the real direct cause and how, whatever the factor it can explain.
Yeah, so I think we're going to do some of that. I've been hesitant because in my experience with gene expression studies is you do anything you want and you see differences in gene expression. Any manipulation, any experiment, and the challenge is sorting through. It's not that you don't get stuff, it's sorting through it. And so each of these regions, it's 2 megabases. There are hundreds of genes in there. Not all of those are expressed in the testes. And so, it might be that we could narrow that down. And the fact that the spermatogenesis gets so far makes me encouraged because it suggests we won't be missing, cell types that are doing a lot of gene expression. So, that is somewhere that we're going, and maybe we should have done that a while ago, and I was just being too cautious. But certainly that would be a way to narrow it down.
Very, very specific.
I think so, yes.
So, yeah, and then I was also shocked to see the sperm head bundles look really okay. Because, pretty much any of this, the latest spermatogenesis phenotype individualization program, always that scatters all the sperm heads. That's our experience.
I see.
The yours are very nicely together.
Stuck together, yeah.
But that, tells us even this, the phenotype is even more specific, which is, to me, very encouraging.
Yeah, so it does seem, and that's another reason why I feel like at least that distal half, it should be the same thing or the same kind of thing, even though they're genetically separable. There's something they're converging on.
Well, thank you, Colin.
Thanks very much.